Hi there, it's me again. This time to bring you up to speed on functional groups, those little constellations of atoms that do different things and that we see over and over again in biology. So here are the 7 functional groups you should know for this course. Sulfhydryl groups. They're important in redox reactions. They can form covalent bonds with each other, thereby cross-linking the macromolecules they're part of. And they're also known as thiols. I think that's what you guys learn in organic chemistry, and can be used to sense the redox potential of the cell. Some proteins with sulfhydryl groups will be in one conformation inside the cell and another conformation outside the cell. For example, in the bloodstream, where there is a different redox environment. So sulfhydryls are a very important functional group. Hydroxyl groups. Anything a hydroxyl group gets added to typically becomes more water soluble. Your liver is very busy right now adding hydroxyl groups to digestive byproducts and toxic metabolites. Any hydrophobic or oily molecule that ultimately needs to be excreted by the kidney needs to become more water soluble, so we add a hydroxyl to it. Methyl groups. Sort of the opposite story. Anything a methyl group is added to is less water soluble. It's a non-polar functional group. And methyl groups are important in gene regulation. The cell even tells old DNA from new DNA based on whether the adenines or cytosines are methylated. Amino groups. Over here. We'll spend more time later with amino groups. They're happily involved in hydrogen bonding, and they are basic, meaning they'll accept protons and become positively charged at physiological pH. Phosphate groups, very important. We need to add negative charges here and here. And adding a phosphate group to a molecule adds a major negative charge. And so that's going to change its shape. One of the major ways enzymes are turned on or off in cells is by adding or removing a phosphate group, thereby changing the shape of the enzyme and rendering it active or inactive. Phosphorylation or dephosphorylation of proteins function as an on-off switch in biology. Phosphates also don't like each other very much. So when we covalently link phosphates together, such as in pyrophosphate or adenosine triphosphate, ATP, with its 3 phosphates stuck together, there's a lot of tension in those bonds, so we can recover quite a bit of energy when they're broken. Carbonyls. We'll talk more about these functional groups in another slide, but suffice it to say they are quite reactive, so we often see molecules reacting at their carbonyls. Carboxyl groups. They're important because they function as weak acids, losing a proton into solution and taking on a negative charge. I'm sure you've noticed that I've been adding these R's to functional groups. Just think of R as the rest of the molecule. Just to make sure, so we still see that there are 4 bonds for each carbon, for example, and then the R is just the rest of the molecule. That's where the 4th bond is. So you should practice drawing these 7 functional groups just as you've been practicing drawing amino acids, and you should start noticing these functional groups as you are drawing the amino acids. You'll see they appear everywhere. Let's look at a couple of these functional groups in a little more detail. As I mentioned, a methyl group decreases the water solubility of anything it's attached to. Those carbons and hydrogens share their electrons equally. So these single bonds all have 2 electrons, and those electrons are about equidistant between the carbon and the hydrogen, at least most of the time. And therefore, this is a non-polar molecule that's not going to form any hydrogen bonds with water or any other polar molecules. Water will actively exclude this methyl group if it can. Meanwhile, hydroxyl groups increase the water solubility of anything they're attached to. So, and that's because they form nice hydrogen bonds. They do not share their electrons equally. That electronegative oxygen there is going to have its electrons right down near the oxygen and pulled away from the carbon or the hydrogen that it's attached to. And that creates a partial negative charge there, and we'll see a nice linear hydrogen bond formed here between the water molecule and that hydroxyl oxygen. Remember, a hydrogen bond is a hydrogen that is shared between two electronegative atoms, such as oxygen or nitrogen. So it's not a bond between two hydrogens, it's a bond that's formed when a hydrogen is shared between two electronegative atoms. In addition to being more water soluble, hydroxyl groups are also more reactive than simple CH groups. A very common reaction that we're going to see over and over again they participate in is something called a dehydration reaction, where essentially a water molecule is removed from the two molecules and they become one single molecule linked by an oxygen. So hydroxyl play an important role in those type of reactions. Amino groups and carboxyl groups readily ionize, which means that the amino group acts like a base and picks up a proton from solution and becomes charged over here. So we've got this charged amino group, a positive charge, and the carboxyl group is ready to lose its proton rather easily. So we can imagine a proton going into solution there, and we end up with the negative carboxylate group there. As I mentioned before, carbonyl groups are more reactive than most groups and are frequent points of attack in biochemical reactions. Two of the types of carbonyls that we're going to be looking at are going to be ketones and aldehydes. This could be hydrogen and this could be hydroxyl, for example, and the same over here. Or well, let's just make this. If this were all H's, for example, that would be acetaldehyde. The key thing to remember about an aldehyde is that this substituent here is an H, so it's less complex than here. And we're going to run into this a lot when we we're studying sugars, because we're going to have ketoses such as fructose and aldoses such as glucose. So you should know the two types of carbonyl that we're going to run into in biology quite a bit: ketones and aldehydes. Instead of trying to fathom the overall structure of a biomolecule, because some of these are going to be huge macromolecules, you should immediately train yourself, or start right now to train yourself to try and spot functional groups instead. Break these molecules up into these groups that are each responsible for doing things. So you should be able to find 3 functional groups in this amino acid. It happens to be a Zwitter-ion right now because one of the groups has ionized negative and the other has ionized positive, but the net charge is 0. I'll give you one hint, and that is that this is not a ketone. So take a break now and go ahead and start drawing some amino acids and functional groups, and just practice. It'll really stand you in good stead.